Hello and welcome to Teach Me in 10, the series brought to you by Technology Networks, where we dive into complex topics and break them down into 10-minute insights. Today we're joined by Chris Tuczemskyi, a chromatography specialist at ThermoFisher Scientific based in Cambridge, UK. Chris joined ThermoFisher Scientific in 2014 and has gained extensive experience in HPLC and water testing. Since 2022, Chris has been part of the product marketing team promoting ThermoFisher Scientific's unique chromatography products. On that note, I'm pleased to say that this episode is kindly brought to you by ThermoFisher Scientific. Their mission is to enable their customers to make the world healthier, cleaner and safer. Without further introduction, let's dive into this episode of Teach Me in 10. Hi Chris, thank you so much for joining us today. So I want to just dive in. Would you mind telling us a little bit about your background and why you're so passionate about HPLC?
Sure. So I guess my background in science in general started when I was a young kid, trying to grow crystals. And really my passion just carried on throughout my school years into university, where I studied biochemistry. But instead of getting a job really in that field after graduating, I ended up in an environmental lab testing drinking water quality. And that's where I first really truly understood the need for quality control testing and ensuring what we consume is, properly tested. And that's also where I was exposed to the power and I guess the importance of HPLC, which is critical in that industry. And then really for the past 11 years, I've been with ThermoFisher Scientific, where I've seen just how integral HPLC is really in ensuring the purity of our medicines now and also future therapeutics as well.
That's brilliant. So you mentioned purity there actually, and let's talk a bit more about impurity analysis in pharmaceuticals specifically. What happens if drugs aren't pure?
That's a great question. Drug purity is really incredibly important as it not only directly affects both the efficacy of drugs but also patient safety. So even like trace impurities can alter how a drug works in the body or can cause harmful side effects really. I guess a good example that best demonstrates the importance of drug impurity analysis is the nitrosamine contamination issue. A number of years ago there were some medications that were recalled due to the presence of nitrosamines, which are really classified as potential carcinogens, so you really don't want to be having them, especially in any medications you're having. So this really highlighted the importance of early and rigorous impurity analysis to ensure compliance with global safety regulations. So yeah, really testing not only ensures patient safety, but also maintains drug quality and also prevents costly recalls as well.
That does definitely seem to highlight the importance of purity. The next one we've got is a bit more technical. So how can you set yourself up for success when analysing samples with strong and weak or absent chromophores?
So this is one of the biggest challenges with impurity analysis, and that's because not all compounds absorb UV light, which means that standard UV vis detection, which is one of the most common types of HPLC detection out there, might not be suitable for this type of analysis. So really, to ensure comprehensive impurity detection, there's, I'd say, a couple of key factors you really need to think about. I guess the first one really is column selection, using a stationary phase that can enhance separation and improve resolution between both the weak and strong chromophores, so you can really get proper detection there. Another, I guess, simple but basic one is the use of internal and external standards. So this will not only help you track your analyte recovery and also help ensure accurate quantification as well. And then finally, really, I guess, with detection, really you need to use modern detection techniques to overcome this challenge of compounds that lack chromophores, really. And by that I mean using an alternative detector compared to a standard UV vis one that doesn't rely on the presence of a chromophore.
Fantastic. So you mentioned there some of the challenges, and what are some of the other challenges that can arise during impurity analysis, and how can detection strategies help tackle some of those challenges?
Yeah, another good question there. I touched on it briefly in the last answer, but I would say really impurity analysis, it comes with several hurdles, but three major challenges that stand out for me. I would say the first one is quantifying unknown impurities. So some impurities it's hard to get standards for, or they just might not be available. And so in such cases, really you want a detection method that provides a universal response that will help you estimate the levels present within that drug or sample. The second challenge I would say would be gradient compatibility. So really using a gradient is a staple throughout HPLC analysis. It helps speed up analysis and helps with separation. But it can also affect the detector response depending which detector is used, leading to inconsistent signals really. So having a system that can overcome this challenge with something like the inverse gradient compensation ability will help stabilize this response. And then I'd say the third challenge really is having a uniform response across all analytes. So some detectors have varying sensitivity depending on the molecular properties of the compounds, which can lead to inaccurate impurity quantification. These properties can include refractive index, light absorption, and also their ability to fluoresce as well. So choosing a detector with consistent response characteristics ensures reliable and consistent data really. So I'd say, yeah, to overcome these challenges, yeah, you really do need a setup that can achieve all of them, giving you greater accuracy, compliance, and confidence with your results, really.
Fantastic. That leads quite nicely into the next question, actually. You mentioned there about having the right detector and the right setup. If someone needs to quantify both APIs and impurities in the same run, how does selecting the right detector maximise the accuracy and precision?
That's really where the choice of detector becomes critical, I think. When we're looking at this kind of analysis, APIs are often present in high concentrations, while impurities, they normally exist at very trace levels. So making dynamic range and sensitivity of a detector really essential for this type of analysis. So when selecting a detector, when you're looking at APIs and impurities, the first thing you'd want to consider is the dynamic range of the detector. I would aim for at least 3 orders of magnitude to accurately quantify both the high-level API and the low-level impurity in a single run. Detection limits as well is very important, so you want a detector that can achieve a good LOD and LOQ, something that's in the low nanogram range, ensuring that even trace impurities can be reliably detected. And then also, I guess the final consideration would be looking at the reproducibility across different concentrations, because some detectors struggle with linearity at the very low end or very high levels, which can compromise your precision when you're looking at those extremes. So an example detector, I guess, that's really, relied upon for this type of analysis is the charged aerosol detector, or CAD, as we call it. As it provides a consistent response regardless of whether or not a chromophore is present, and this kind of detector really makes it easier to analyze APIs and impurities together on the same single run, even when reference standards aren't available.
Fantastic. That seems like a great summary, Chris. Finally, are there any other tips that you might have for our audience that we maybe haven't discussed?
I think I've got a couple that come to mind, really, I guess. If we look at everything as a whole. I guess the first tip is really to use complementary detection techniques, so not just relying on a single detector such as UV vis, but combining your classic UV vis detector along with a CAD or charged aerosol detector, but also a mass spec which would really help provide a complete impurity profile and give you that confidence in your results, really. Other techniques that are becoming more popular as well is looking at 2DLC techniques. So if impurities collude with APIs, using something like 2DLC can help improve separation and help reveal those hidden compounds. And then, I guess a more generic one, but also very important, is really choosing the correct hardware and also the consumables. So making sure you use high quality vials, columns, and also choosing the right system. All those kind of things will help contribute and help you achieve consistent, reliable results day in, day out without any issues.
Fantastic. Thank you so much for those insights, Chris, and thank you very much for joining us today and give us some incredible discussion points. That wraps up today's episode of Teach Me in 10. A huge thank you to Chris Tuczemskyi for sharing his expertise on impurity analysis in pharmaceuticals. We hope you found this episode enjoyable, and be sure to keep your eyes open for our next episode of Teach Me in 10. Until next time, thank you for watching.